Hey, my name is Morten Allehud. Welcome to this session about biofilm properties. There are many definitions of bacterial biofilms, but the most common definition is an aggregation of bacteria on a surface embedded in a self-produced matrix. In order to understand biofilms, we have to study them, and there are indeed many ways to study biofilms. In vitro assays vary from very simple and cheap models to complex, but also more realistic models. In the simple end, we have the crystal violet assay. This assay is very cheap and simple biofilm model. The assay is assessing the quantity of attached bacteria in the walls of a plate, but it is not discriminating between live and dead bacteria. Another simple way to study biofilms is the filter biofilm. Here the biofilms are grown on top of a micropore filter on top of an agar plate. The filter enables transfer of the bacteria onto fresh agar plates when needed, and hence the bacteria can be supplied with fresh nutrients, allowing the biofilm to form. A third model that we have developed here at the Department of International Health, Immunology and Microbiology, we call it the semi-solid model. Here the biofilms are not attached to a surface, but instead kept in a semi-solid matrix composed of digested meat powder, horse blood, and serum. The idea of this model is to provide a more realistic biofilm model where host components are included. The importance of host components is dual. Firstly, the formed biofilms are different to biofilms raised in simple media, but secondly, the host components effectively bind antimicrobial molecules and hence must be considered when evaluating antibiofilm treatments. There are, as mentioned, numerous of biofilm models. However, the gold standard has always been the flow cell systems. This system utilizes a continuous flow on top of the bacteria growing in a chamber like this. The idea is to keep the bacteria supplied with a low but constant supply of nutrients and flush out non-attached bacteria, and hence to form biofilm attached to the bottom of the chamber. Many extremely beautiful images and videos of biofilm formations have been produced using this assay. From a distance, it looks like a simple stepwise process. First, the bacteria attaches to a surface, then they grow into huge towers looking like mushrooms, and lastly, the bacteria within the mushrooms disperse into the surrounding media to form new biofilms and colonize new surfaces. However, let's take a closer look into the mushroom-looking towers. First, the stalk is formed by clonal growth, meaning that the bacteria divide and multiply to form the stalk. In contrast, the cap is dependent on motility. See here how the motile bacteria crawl on top of the biofilm to form the top part of the biofilm. During this stage, the so-called matrix is produced. The matrix is a slime substance composed of mainly water, but also on all available macromolecules, such as polysaccharides, DNA, and protein. These structures are meant to be interspersed by channels to ensure nutrient supply and waste disposal. However, when measuring oxygen concentration within a biofilm, it is obvious that the supply is not sufficient. The oxygen is rapidly consumed by bacteria in the outer layers, and subsequently, the middle part is almost depleted from oxygen. Due to this lack of oxygen and possibly also other nutrients, the growth rate within a biofilm is dramatically lower than the planktonic counterparts. The matrix components, the lack of oxygen and subsequent the slow growth, are all contributing to the hallmark of the biofilm, namely extreme tolerance towards antimicrobials and immune cells. Due to these factors, the biofilm can simply resist doses of antimicrobials that would otherwise be lethal to the bacteria. Recall that antibiotics mostly target metabolic processes, so if they once penetrate the matrix, there are no targets to inhibit. Interestingly, it is only physiologically dependent processes that make the biofilm tolerant. Therefore, it is not a genetic or inherited trait, as is antibiotic resistance. Hence, all traits making the biofilm tolerant are reversible and can be circumvented. However, are these matrix-embedded mushrooms found outside a flow chamber? Are they found at the site of chronic infections and thus being important? So I guess the important question is, are flow cell biofilms a realistic model that resembles what is going on during infections? In a recent publication, we looked into that question. Interestingly, we found that biofilms found in the body are actually very small, ranging from only 5 micrometers to 100 micrometers. And none of them looked like the enormous mushroom structures we've seen in flow cells. In addition to being very small, the biofilms are often very heterogeneously distributed, meaning that they can be extremely hard to find and diagnose. Another interesting observation was that even though biofilms are per definition attached to a surface, it has been shown that aggregates are equally tolerant to antimicrobials and immune cells as surface-attached bacteria. This conclusion is important since many in vitro assays are evaluating the antibiofilm properties with surface attachment as an endpoint. But what is the compound is just releasing the tolerant aggregate into the bloodstream? This stresses the need for more realistic biofilm models. So to sum up, we know that the biofilms are aggregates of bacteria embedded in a matrix of various compounds. The hallmark of the biofilm is to be extremely tolerant to outside threats. And this tolerance seems to be reversible and thus dependent on physiological properties rather than genetic and inherited traits. And that is why biofilms is not a part of the resistance problem we are also facing in our environment. Biofilm needs its own research. Biofilms can be studied in numerous of different models in the lab, but it is very important to know that all models have drawbacks and no models resemble what is found in nature.